Okay, so in the last class, we were talking about uh, some of the things that have been discovered ever since the modern synthesis crystallized in the early 1950s. And we started with asking about or talking about the concept of epigenome. Okay, so what exactly is the epigenome? So, what we'll do is we'll first start by looking at what is the concept of epigenome. Okay. Mm, okay. Where is huh, this is the one. Yeah. Can everybody see the screen, people?
Yes.
Yes, sir.
Okay.
DNA contains the instructions for building all the parts of the body. But DNA is only half the story. The DNA in our bodies is wrapped around proteins called histones. Both the DNA and histones are covered with chemical tags. This second layer of structure is called the epigenome. The epigenome shapes the physical structure of the genome. It tightly wraps inactive genes, making them unreadable. It relaxes active genes, making them easily accessible. Different sets of genes are active in different cell types. The DNA code remains fixed for life, but the epigenome is flexible. Epigenetic tags react to signals from the outside world, such as diet and stress. The epigenome adjusts specific genes in our genomic landscape in response to our rapidly changing environment.
Okay. So, right. Mm, So now I have to stop presenting. Yeah. Now the presentation goes to enter. Okay. Huh. So this is what we were talking about yesterday, Richard Dawkins' selfish gene, which essentially tries to say that all the features of an organism are chiefly determined by the genes, and therefore the implication was that environment was merely an agent of selection. It is in this context that we started talking about the epigenome, and the epigenome is essentially the complete description of all the chemical modifications to DNA and histone proteins, chemical modifications which do not alter the ATGC sequence. This is a very important part that regulate the expression of the gene within the genome. And since we know that the spatial and the temporal pattern of gene expression are very important in terms of figuring or determining the phenotype of the organism, therefore it's easy to see how the epigenetics and the sum total of the epigenetic changes, which is the epigenome, how that ends up or how that can potentially end up affecting the traits. So in this context, what I'll show you guys is a very interesting trait. So, okay, this is refusing to play, doesn't matter. So essentially, have you ever seen a rat mother taking care of its babies. If you have seen them, which is what this video was all about, then you will see that there are two particular behaviors that apart from feeding them obviously, that the rat mom keeps on doing over and over again. So one of them is it licks them with their tongue all over the body. And the other thing is that it keeps on, just like this, it keeps on grooming the babies. Now, in humans, mostly human mothers don't lick their babies, but this grooming behavior, okay, continuously just stroking or petting the baby is something that is found even amongst human moms. Now, turns out that there is quite a bit of variation among rat mothers in terms of how much licking and grooming they do. So this behavior is known as licking grooming behavior. So there are some rat mothers which tend to do a lot of licking grooming for their babies. These are the high licking grooming mothers. And there are other mothers who kind of cheat a bit. They don't do too much of licking and grooming. Now, turns out, very interestingly, that the behavioral phenotype of the offspring of the rat baby depends critically on what is the nature of the mother. So if the mother is just one sec. So if the mother is, say, a low licking grooming kind of a mother, then the baby ends up becoming an anxious adult. Okay, so if you take such rats and if you, for example, leave them in a new arena, so rats have this behavior that normal rats, they will go and explore the entire arena. But these rats, they won't do that. They will just sit in one corner and kind of, keep cowering in one corner. Similarly, if you try to, hold a rat by its tail and you try to drown it in water, then obviously, a normal rat will not like it. It is going to try to, arch its spinal cord, and it tries to escape the drowning as much as it can. On the other hand, if you take some of these anxious rats and you try to drown them, they won't do much. They will basically happily just get drowned. So that's what I mean by anxious. Okay, so these pups which are raised by low nurturing mothers, low licking grooming mothers, these pups tend to be anxious, and those pups that are raised by relaxed, high nurturing mothers, they end up becoming relaxed, normal rats. Okay. Now, obviously, if you get a scenario like this, then there are two ways in which this can happen. One way is that there is some genetic component. So mothers which are anxious and low nurturing themselves, they pass some gene to their pup, and that gene is what ends up making them anxious. On the other hand, if you have these high nurturing mothers, they pass a high nurturing gene. The alternate possibility is that it is nothing to do with the genes. Both rats have similar genes, but it has something to do with the act of raising the babies. So something in the act of raising the babies, that is what is making these guys, this set of babies anxious and this set of babies normal. So how will you distinguish between the two cases, which is which? So the simplest way of figuring this out is if, let's say, you take sets of biological mothers, some which are relaxed, normal, some which are anxious, and then you take a bunch of foster mothers. So these are mothers who are not biological parents, but they are the ones who raise the baby up. So essentially, you have two kinds of foster mothers, those who are relaxed and those who are anxious. So now you have a 2 into 2 combination, 4 combinations. So this cell is where the mother is biological, the biological mother is not anxious, is relaxed, and the foster mother is also relaxed. Biological mother is anxious, but foster mother is relaxed. Biological mother is relaxed, but foster mother is anxious, and both biological and foster mothers are anxious. So those are the 4 possible combinations. So you do it in those 4 possible combinations, and if the experimental data will look like this. So essentially what it says is that the identity of the biological mother will not matter. It is the foster mother's identity which will determine whether the offspring is relaxed or whether it is anxious. Okay, so this suggests that a very important and potentially fitness determining parameter like anxiety, etc., that of the offspring is not entirely genetically determined. There is a massive behavioral component to it. So then obviously the question is, how does it work? How is it that the simple act of licking grooming can, that too in the early life, that can lead to such a massive change later on in the life? So like most things in biology, the answer is pretty complicated. There are lots of pathways, loops that are acting, but overall the main thing which has the maximum contribution to the phenomena looks something like this. So in the brain you have a region called the hippocampus. Okay, now in this hippocampus region you have certain proteins called glucocorticoid receptors, which are found on the surface of the brain. Okay, now typically whenever a mammal gets anxious, then what happens? Then you have the synthesis of the flight and fight set of hormones, right? The adrenaline and the cortisols, basically from the kidney, or rather not from the kidney, from adrenal, or the gland near the kidney. Now these hormones, they obviously have a profound change in the behavior. They are the ones which allow you to cope with a stressful moment with very high degree of alertity, etc. However, once they have fulfilled their job, then this needs to be removed from the blood. If it is not removed from the blood, then you will be in a perennial state of heightened awareness, and essentially which will show up as anxiety. You'll be in a perennial anxious state. Now in order to remove this, the body transports these proteins, the cortisols, through the blood to the brain. And in the brain, it is these glucocorticoid receptors which actually take those proteins and essentially pull it inside the hippocampus, thus reducing the protein level in the blood, in the body, below the threshold, the threshold of anxiety. Therefore, the number of glucocorticoid receptors in the brain is very important in determining the anxiety phenotype of an adult. So if there are more number of glucocorticoid receptor in the hippocampus, then the cortisol levels can be brought down fast, and therefore there is less anxious behavior, and vice versa. Now turns out that when a baby is born in rats, then the receptor for the promoter for the hippocampal glucocorticoid receptor gene is highly methylated. Okay, so what is meant by highly methylated? So that means that the promoter has a large amount of methyl groups stuck to it, as a result of which the transcription of that gene is not possible. Now when the mother does the licking grooming behavior, and this we are talking about a particular phase, I think first 1 week or 10 days or so in the early life of the baby. So during this period, when the mother does all this comforting by licking grooming, then a series of reactions are set, because of which these methylation marks are removed. And when these methylation marks are removed, then so this is particular methylation level of that thing, and this you see that when the mother is low nurturing, then the methylation level remains close to 100%, whereas when raised by a high nurturing mother, the methylation level comes down to 10% or less. Okay, so essentially, this nurturing greatly ends up reducing the methylation of the promoter, which increases the expression of the GR gene. And once the expression of the GR gene is expressed properly, then there are a number of those receptors in the hippocampus. And then this is what, in the adult stage, makes the offspring a relaxed adult. Now, very interestingly, this bit is important only in the context of the early life. So there is a critical period after which, if you do a lot of licking and grooming, it will not matter, because the number of GR proteins in the hippocampus region is set in that early life period. Now, very interestingly, although we studied this in the context of rats, it is known that this particular thing can have a possible role even in human beings. So what they did was a very interesting study on the effect of childhood abuse on the epigenome. So what they did was, for this, remember, they need to take brain sections, and obviously it's hard to get brain sections of human beings. So what they did was, they went to the morgue, and then they got hold of those bodies where people were ready to allow them to cut brain sections. And then for these, they tried to figure out the early childhood history of those dead people. Okay, now obviously that's not very easily done, but still they kind of ended up getting a reasonable sample size for this, about 12 in each. So they then divided these samples into three types. Okay, those people who had committed suicide and had an abused childhood, those people who committed suicide but there was no record of abuse during the childhood, and control people were those who had died of natural reasons, not because of suicide or anything. So the idea over here is that if somebody is committing suicide, then they are assuming that that person is or was in a highly depressed, unnatural state of mind. And what they are trying to see is that whether that is something that ends up having so that is the condition of suicide is what they are taking as a proxy for whether the person is suffering from severe mental trauma or severe mental depression or not. And then what they're really trying to see is this comparison on one hand and this comparison on one hand. So these people, both these groups, they are childhood non-abused, but this group is suffering from mental trauma or mental depression, and this group is not. And yet their methylation levels of the gene which is analogous to the glucocorticoid receptor in humans, the methylation levels are not significantly different. However, if you compare this group, both of which have committed suicide, therefore mentally, hopefully both of them were in a bad state. In both the cases, in these cases, you can see that those who are abused, they have much lesser level of glucocorticoid. This is the expression level of total glucocorticoid receptor mRNA, and this is the actual level of the glucocorticoid receptors in the brain. And now if you look at, for the same groups, if you look at their methylation levels of these genes, you see that these guys have far greater methylation levels on an average compared to these guys. Okay, it's still not the 100%, 10% as I showed you in rats, but still this is like 4 times, roughly speaking, of these guys, right? So which essentially says that humans who have an abused childhood, they end up showing more methylation of the promoter for the GR gene, which, based on what we understand from rats, is probably what is responsible for this kind of a depression. Interestingly, a similar phenomenon, it has been shown not in the context of methylation, but in the context of effect of early childhood abuse on personality later in life. So, have you guys heard of the Romanian babies? Anyone? How many of you have heard of Romania, the country of Romania?
I have.
I have.
Okay, cool. So roughly, where is it?
Eastern Europe.
Yeah, it's
Okay, fair enough. Fair. So this country in the 80s was ruled by a dictator called Nicolae Ceausescu. Now, Nicolae Ceausescu, he had this thing that he wanted to revive the economy of Romania. It was a poor country. It still remains a poor country by and large. And therefore, he was under the opinion that they need to have a very large population so that they have a very large workforce. Therefore, Nicolae Ceausescu ended up banning abortion in Romania. Now, of course, when you ban abortion in a poor country, then life becomes very difficult. Human beings didn't change their behavior, but essentially there were a large number of unwanted babies. Okay, and these babies were being abandoned regularly in the hospital, in the church, near bus stand, pretty much everywhere, garbage dumps, etc. Now, since there is a government policy, or there was a government policy that the babies cannot be aborted, therefore, these babies, you just can't kill them. Anyway, you can't kill them, but in that country, it was a bigger thing. Therefore, Romania ended up building massive orphanages for housing these babies. And the condition of these babies in those orphanages were absolutely, absolutely terrible. So they put like 8 or 10 babies in one small baby cot, that kind of stuff. And obviously they didn't get very good food. And most of the time there were very little caring that was happening for those babies and so on. So in 1989, there was a coup in Romania and Nicolae Ceausescu was shot. So when that happened, then the plight of the Romanian babies became known to the outside world, and several childless couple or good humanitarians, they ended up going to Romania and taking or adopting these babies and bringing them back to the first world. So these babies, they are the so-called Romanian babies. When they grew up, when they became teenagers and slightly later than that, then it turned out that almost without exception, these babies are suffering from certain personality disorders. So for example, most of them, they cannot form emotional bonds with anyone. They cannot become empathetic to someone. Okay, so basically, see, when suppose some near and dear people to you, suppose somebody falls down and has a pain somewhere, then even if you are not feeling the pain directly, you end up feeling for that person. Okay, you feel bad about it. That's sympathy. So these guys, they cannot feel empathy, and they are again perennially anxious. They become agitated very easily, and so on and so forth. So essentially, they were behaving like the human analogs of the rats. So there is a very strong hypothesis that these Romanian babies probably are anxious precisely because of this, what you are seeing on the screen. Essentially, they have very little glucocorticoid receptors in their brains, and those glucocorticoid receptor genes are very highly methylated in their brains. Now, obviously, you can't just take those babies up and cut their brains open. So people are still waiting for some of these guys to die, hopefully of natural reasons, and see whether this hypothesis is borne out in them. Although the chances of that happening, based on our understanding coming from quite a bit of rat work like this, seems that the chances of that happening are pretty high. Okay. So coming back to evolution. So if you remember that in modern evolutionary synthesis, the idea is that the genes are the primary features that are determining the features of an organism, and the role of the environment is simply to act as an agent of selection. However, now based on what we said, we realized that at least some of the features of the organism are determined by an interaction of the genes and their corresponding epigenetic markers. Therefore, and what epigenetic markers will come is obviously going to depend upon what environmental conditions the organisms end up facing. Therefore, environment is no longer a simple agent of selection. Environment, now we understand, is also playing a role in terms of determining at least some of the features of the organism. Now, unfortunately, if we take this thing seriously, then there is a slight problem. What is the problem? The problem is that in order for all this to happen, the changes that we are talking about, these changes must be inheritable. Remember, I said that in order for natural selection to operate, evolution by natural selection to operate, at least part of the trait has to be inherited from the parents to the offspring. If that doesn't happen, then evolutionarily speaking, that change is not making much sense. However, we know for a fact, courtesy this man, August Weismann, you know what he did, right? He took mouse, and for several generations, I think 19 generations or so, he cut their tails. And he was trying to see whether the babies that are being born, whether they are born tailless or not, because of the acquired character of getting the tail cut. But even after 19 generations, he found that the babies are still being born with tails, which, if you think about it, should not have come as a surprise of all people to August Weismann, who was a Jew. And you know that Jews, like Muslims, the males at least, they undergo circumcision. And the point is that the reason that you need to undergo circumcision every generation is precisely because when the babies are born, every generation, they still have that foreskin. So therefore, in some sense, we human beings have been doing Weismann's experiment for the last at least 2000 years in the context of Jews, and at least the last 1400 years in the context of followers of Islam. But all that information essentially tells us that whatever characters that are acquired are never going to be inherited. Now, in the context of epigenetic modifications, we know that when the process of gametogenesis happens, okay, gametogenesis is the process by which gametes are formed, sperms for males and ova for females. So when that process happens, then there is something known as epigenetic reprogramming that happens at the level of the cell. Essentially, the genome is stripped of all the markers that lead to these epigenetic modifications, and that is the reason for which acquired characters are so hard to get passed on to the next generation, even when they are operating through the epigenetic markers. So if that be the case, then it seemed that epigenetics was a very nice phenomena, a sexy phenomena, but it seemed that it would not have any role to play in the context of evolution. However, I mean, this is the stuff. Acquired traits cannot be inherited, and therefore adaptive evolution occurs through the differential survival of competing genes alone. Okay, nothing acquired has a meaning. However, one of the things that we have figured out over the last 10-15 years or so is that the epigenetic reprogramming that I talked about during gametogenesis, that is actually not a foolproof thing. So there are quite a few epigenetic changes which can be transgenerational, which means they can be passed from generation to generation. So for example, if you take a mouse and a pregnant female, and if you end up hitting it with this drug called vinclozolin, then you will see that the methylation pattern of the genes gets altered in this female. Okay. Now, if you look at the babies that this female is producing, then you see a bunch of abnormalities in them, including things in the testes, in the prostates, problems with the kidneys, immunity, etc., etc. Interestingly, if you now take these rat pups and make them breed, then for at least 4 generations, all the male progenies, this is very interesting that these abnormalities are only seen in the males and never in the females, but at least for 4 generations, all the male progenies end up showing all these diseased phenotypes. Now, please remember over here that in this experiment, the pregnant female is being hit with vinclozolin only once. After that, in the future generations, there is no vinclozolin treatment that is being given. So this was one of the cases which ended up showing rather drastically that at least some epigenetic changes can pass from generation to generation. Another classic example of this comes from the plant kingdom, where you have this funnily named plant called toad flax, scientific name Linaria vulgaris. Now this Linaria vulgaris, so I mean, okay, this word vulgaris comes from the word vulgar. Vulgar nowadays has a meaning which is kind of close to gross or anything. Okay, uncouth, uncultured, etc. However, the word vulgar in its original form actually simply meant common. Okay, so Linaria vulgaris is literally common Linaria. So in this particular plant, the flowers, it was known that they came in 2 forms. One form was this thing which was had 5 finger-like stuffs, radially symmetrical. This is what is known as fine. So one form was this one, Forma pelorica, and the other was called Forma typica, in which the symmetry of the flowers is bilateral, not radial. Okay. Now, for a long time, people thought that this is happening because of 2 different alleles in the plant, because these guys bred true and these guys bred true. It was very stably inherited thing. Now, in this is the typical form, this is the more common form, and this is the form which was rarely found. And this form was thought to be a mutant. However, when sequencing became more popular, they actually figured out that both these plants, there is no difference at the level of the genes. Now, in plants, the flower morphology was supposed to be handled by this gene called L-Sic, and they ended up, when they did epigenetic sequencing of these, they found that this L-Sic gene of this form is actually excessively methylated, and interestingly, this methylated pattern is very, very stably inheritable across generations, which is why people were thinking of the whole thing as genes. Now, when they studied it in detail, they found something really fantastic. Okay, so remember, this is the typical form and this is the methylated form. This is unmethylated, this is the methylated. So when they looked at this plant in detail, they actually found that you not only find these 2 extremes, but you also find these intermediate forms. Okay, and in these intermediate forms, you can see that the length of this spar, that is indicated by this arrow, the length of this arrow, these spars are greater. So what they did was they arranged these flowers in order of increasing spar length, and then they actually checked at the methylation levels, and interestingly they found that this has the least one, this has the most, and then the methylation level actually increases as you go from left to right. So essentially, this is not a simple on-off qualitative Mendel kind of a scenario. It's a quantitative thing. How much methylation is there is what is determining what is the length of this spur, and therefore what is the exact phenotypic form of the spur. Okay. So ever since, there has been huge number of examples over the last 20-30 years which have shown beyond a doubt that there are several epigenetic changes which actually escape the epigenetic reprogramming during gametogenesis. So coming back to evolution, what is the implication for evolution for this? So what it suggests is that it's not that acquired traits cannot be inherited at all. At least some acquired traits can be inherited, and therefore adaptive evolution, remember the modern synthesis was talking only in terms of the genome and nothing else, but now epigenetics says that adaptive evolution can potentially happen at both levels, the level of the genome as well as the level of the epigenome. Okay. So the third very important concept over here is that it is assumed, because you adopt the biological species concept, that all genetic information is passed within the same species. And that is why when you look at the trees, okay, the phylogenetic relationships, why are they called trees? They are called trees because they have this kind of a branching structure. And the primary implication of this kind of a branching structure is that any given branch, any given twig will have exactly one and not more than one ancestor. Okay, two twigs or two branches can have a common ancestor, that is fine. But a given branch can have one and only one ancestor, and that is what is behind Darwin's conceptualization of descent with modification, forming this kind of phylogenetic lineages. That is what leads to this kind of trees. However, one thing that is becoming clearer and clearer as we look at the genomes of more and more organisms is that this is something that doesn't seem to be true at all. So a poster boy example of that is this particular species of slug known as Elysia chlorotica. So this particular species of slug, this is the only photosynthetic animal. Okay. However, when it was born, it's not green colored like this, it is brown colored. And what it does is it ends up eating a particular algae called Vaucheria. But it doesn't digest the whole of that algae. What it does is it retains the chloroplasts. And these chloroplasts keep on accumulating inside the body of this slug till the entire slug turns green like this. Okay. Now, when that happens, then this particular slug, it now stops feeding and lives entirely on the carbohydrates that are synthesized by the plastids. Till this point, there is not a problem, okay, because no genomic integration has happened. However, turns out that plastids require several nuclear proteins to function. And these people had shown that about 52 genes of the algae have become stably incorporated into the genome of the slug, and these proteins or these genes are the ones which are supplying the nuclear proteins for the slugs to operate. Okay, so this was supposed to be like the example of horizontal gene transfer in animals. However, turns out that recent evidences have been very strongly doubting this particular claim. So turns out that there was some major problem in the sequencing that these guys had originally done, and therefore these 52 genes that they seem to see in the genome of the slug, that data is questionable. Then the question is that if that be the case, then how are these Vaucheria plastids able to survive and photosynthesize inside the slug? So turns out that the Vaucheria plastids are of a very different type compared to the plastids of normal plants, which require these nuclear proteins. Vaucheria plastids do not. So Vaucheria plastids can actually essentially be autonomous. And because of that, they don't really need any help from the slug genome. So this particular example, the reason I'm quoting this example to you guys is because when you start seeing, researching about HGT, you will find the example of this slug coming up again and again. But in reality, this slug is no longer an example of HGT. The other poster boy example of HGT is supposed to be this rotifers called Deloitte rotifers, and in this, these guys have been reproducing parthenogenetically, no sex, for the last more than 80 million years. So it's a very ancient species. And the question that people have been asking is, how is it possible? Because we know that if you don't have sex, then there are several negative things that can happen to your fitness. So what they had ended up showing was that 8 to 9% of all their genes have come from other species through horizontal gene transfer, and these 8 to 9% of genes actually represent 39% of all the enzymes that are there in their pathways. Okay, which basically suggests that these guys are essentially replacing sex with horizontal gene transfer. They're getting their extra genes from outside. However, this is really funny. In 2000, by the same group, the group that had said this, saying that the original place they had messed up very, very heavily, and there was actually a lot of cross contamination in that work, and that is what explains all their results. And now there is no reason for them to believe that there is horizontal gene transfer. The interesting bit is that these guys published the original flawed paper in a very big journal, and when they are trying to say that, hey, look, we messed up, they published it in an equally big journal. Okay. Whereas if for certain, which tells you that this is a very big group, because otherwise certain other groups, if they made mistakes like these, they would be like forever banished from scientific community, so to speak. Anyway, now these two examples that I have shown you, these two examples are actually poster boy examples, which is why I'm showing you, just to make the point that these two examples are not correct. However, this is not to say that horizontal gene transfer doesn't happen. Horizontal gene transfer, the phenomena is extremely strongly documented in many, many organisms, particularly in the context of microbes. Horizontal gene transfer is known to be very, very common, and horizontal gene transfer is supposed to be the primary reason for which microbes end up becoming, getting multidrug resistance so soon. So basically what is happening is, suppose you are treating one set of microorganisms with some kind of drug, those guys develop that resistance, and then they pass on that resistant gene to another set of bacteria or microbes which have never been exposed to that antibiotic. So even though you are not using some kind of latest antibiotics to say treat diarrhea, the diarrhea bacteria can still get the resistance to those antibiotics, and therefore your next diarrhea might become very, very hard to cure. So this is something that is happening. Okay. So what is the implication of this? So when you have that kind of no horizontal gene transfer, then all genes pass only from parents to offspring. You get this kind of branch trees. However, now when you have genetic information being passed across species, then the concept of a lineage is no longer well defined, and you can now have these kind of trees where, can you see these branches, these like bridges connecting two branches? So this is what is known as a reticulate pattern. Okay, this is the kind of pattern that you find in veins on leaves. And now technically what happens is, a single branch can have multiple ancestors. Okay, a single branch can arise from multiple nodes, which means the concept of ancestry itself becomes slightly more problematic. Now, how problematic an issue this is. There are certain people who will look at this and say, wow, Darwin is gone, entire evolutionary biology is gone because no descent with modification, trees look like this. However, there are other people who will say that, look, this is something that is, it changes how we view these kind of trees, but it doesn't necessarily change too much of the rest of what we understand about evolutionary biology. It simply changes, our understanding of how to do our taxonomy, phylogenetic taxonomy, but it doesn't change much beyond that. And that is true to some extent, because even in cases where horizontal gene transfer have happened, we see that only a few genes have been transferred horizontally. So the majority of the genes are still being transferred from parent to offspring. It's only a few genes which they tend to acquire now and then from here and there. Now, if that were to be the case, then can we treat horizontal gene transfer simply as something that slightly complicates the picture versus horizontal gene transfer as a complete game changer? Depending on which Pandit you are talking to, you will end up getting both kinds of views in the literature. So, what are the implications for the process of evolution for this kind of epigenetic changes? So, typically genetic changes, they arise by mutations, and by and large the rate at which they arise is reasonably slow. However, epigenetic changes can happen much faster. These are relatively more stable. These are much more dynamic, which means that even if you find them today, they might be transgenerational, but they might end up getting out within 3-4 generations. There is a lot more variation over here in this. Okay, there is less variation here, and because they are rare, the probability of multiple occurrences happening in multiple organisms simultaneously is much lower, whereas the probability of affecting multiple individuals simultaneously is much higher. So, and here is the point, note that an organism doesn't have to do either this or that. For an organism, both kinds of changes are available to them, whatever works for them to increase their fitness. So a lot of work is now happening about trying to figure out the relative importance of this versus this. Nobody is saying that only one of them exists. That's not true. But essentially, people are trying to figure out under different conditions what is the relative importance of this and what is the relative importance of this. Okay, so the take-home message is that epigenetic changes and horizontal gene transfer, they greatly increase the amount of variation that is available for evolution. They complicate the pattern of inheritance because it's not entirely sure whether the offspring will get it from their parents or not. And even if they get it, then for how long in the lineage they will keep on getting it. And therefore, all these, they end up making evolution a lot more of a dynamic process than what is portrayed by modern synthesis. And they have all kinds of practical implications in terms of evolution of drug resistance, invasive species, blah, 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 blah. However, if you look at the theory of evolution, then interestingly speaking, none of them have a major impact on it. They might have a little bit of horizontal gene transfer, might have a little bit of impact on how you make your trees, but epigenetic changes in particular, people have shown that the existing population genetics theory can very easily incorporate epigenetic changes into it without doing any major alteration to the structure of the models. So this is pretty much it. There are the readings one can figure out. So yeah, I think I have kind of overshot the time slightly. So what I'll do is I will now stop the recording, and yeah, so the class is it's coming to my thing. Good.